Kiran and Samia, you're still there, right? What was the last thing I said? With respect to this? That's you cross them. Okay, so at some point you're gonna have cross them during analysis one. We have cross them. Cross them will be some genetic variation. Right? So meiosis mainly occurs in gametes, right? Or to produce gametes. And when we say gametes, we mean sex cells. And so in humans, most mammals have sperm cells and ovum. Right? So most of these cells, well, it depends on male or female, they produce and sperm cells or ovum. And the significance of that is that in humans, right, we're producing haploid numbers. Right? So each one of these is a haploid number, meaning 23 chromosomes. Right? It depends if it's male or female. Like when it meets another, gamete, these 23 chromosomes of the haploid number combine together again to give us a 46 chromosome of two element organs. I always call that first cell. Exactly. And so that's the next significance of meiosis. It maintains the two element diploid. Right? Not in the meiosis state itself, but after fertilization. Okay? Remember, these were 46, the organisms produced would have been 92. Right? In humans, and you don't want that. Right, so I go through the actual process itself. Right, so again, you start off with interphase. And in interphase, we have our nucleolus, the nucleolus membrane, and the DNA is not visible. The chromosomes are not visible as yet. Right? In early prophase, so going from interphase to early prophase, we've seen that the nuclear envelope, the nucleolus envelope is beginning to disintegrate. Right? Or the nuclear envelope, yeah, beginning to disintegrate, and the chromosomes starting to become visible. Right? But it's still not visible as yet. In late prophase is when your chromosomes become visible. Okay? Your chromosomes become visible and they form something called bivalents. Okay. And next significance in any diagram is with your centrioles. From your centrioles is where spindle fibers attach, and then it can pull apart the chromosomes. In late prophase, your centrioles move to the opposite ends of the cell, right? At the ends of the equator. Right? Right? And your bivalents form. Okay. So that is prophase, right? And the main takeaways from prophase is the visibility of the chromosomes, the centrioles moving apart, and the disappearing of the nuclear membrane. The next big significance is this is the stage that crossing over occurs. Right? So we could see that the chromosomes they line up against each other, right? So what would happen is we're looking at the chromatids. So in my diagram, this is interphase, these are improvements. Right? That crossing over could only occur between homologous pairs of chromosomes. Right? They have to be the same length, same size, same everything. So between these dashes, so we can differentiate between these two chromosomes. Okay, so what's going to happen is they're going to line up next to each other and they're actually going to be touching. And what could happen is, now you say this branch lines up with this branch could detach and attach to this one, and this one could attach to this one. So after crossing over, you'll have something like this. The point at which they intersect and exchange information will come up and replace with the other side. Let us see that this piece of the chromatin was originally from here, right? Indicated by the stripes. It originally didn't have any stripes. But the stripes was exchange between chromosome 1, chromosome 2, and that is how this piece became striped. So this indicated that this piece of the chromatin was originally from them. And this blank piece of the chromatin was originally from here. So this is the cytochrome, and this is the genetic variation. Right? And this occurred in late prophase. And the process is called crossing over or recombination. Right? So it's crossing over or recombination, and the products of recombination are called recombinants. Okay? After recombination, you produce recombinants. And you have a specific name here, point at which they cross over, right? So if they cross over at this point, so obviously they'll be touching each other, right? I can't just show. To the point at which they recombine, cross over is called chiasma. Right? So it's chiasma or chiasmata. Right? Chiasmata is plural. It depends on where it goes. So for example, if this chromatin crosses over this one, then these two will exchange. Right? I just say that these two are
Also, you have them in anyway.
yeah, right. So once it's remember they are along the same localizer. Yeah, but in the diagram they show you, it will have where it goes. But remember, the chromosomes they line up at certain points. Right? They'll show you how it lines up. Or they'll just ask you to draw it. When you're drawing it, you can draw it anyhow. So the top could cross with the top, and the bottom will cross with the bottom. Right? Right, so cross, the main thing in prophase, if the most significant step in prophase for meiosis is that crossing over process to form recombinant chromosome. Right, that it's only homologous chromosomes that could go through this process. Right, so we finish with prophase and we're moving on to metaphase. Okay, and the most significant process in metaphase is the lining up along the equator. Right, and attachment of spindle fibers. So at this point, you have no more nuclear membrane, right? And your chromosomes, your genetic information line up the equator, right? Or your bivalents, we call them bivalents at this point. Right? They line up along the equator, spindle fibers attach, and all of that occurs in metaphase. So in metaphase, this is the only phase in which you see the chromosomes line up along the equator. And it have two ways you can line up along the equator, right? It could be vertically, or it could be vertically, or it could be horizontally, right? Because you have two ways in which you can have an equator. Right? And the next step is anaphase. Now, in anaphase is where you're pulling them apart. So you have two stages to anaphase and meiosis. Okay, stage where it's pulling apart. So they're being pulled apart, and later anaphase is when they're already pulled apart to the end. Right? The main point of anaphase is separated to opposite ends of the poles. Right? And then you have telophase, This is where cleavage begins and separation of the singular cell into two different cells. Okay, so the key parts of meiosis one, prophase, the biggest thing is crossing over, right? And condensation of condensation or coiling of the chromosomes. Those are the two most significant events. Right? You have other things, but those are the most two significant ones. Metaphase, you have the alignment along the equator of the chromosomes. Anaphase is the pulling apart of the chromosomes. And telophase is the separation into two new cells. Right? And then there's the end of meiosis one. Right? Anybody have any questions on meiosis one? So at this point, we have two cells, right? But we still don't have a haploid number. Okay? We only have two cells. So if you look at the diagram on the board, this is what the diploid number of chromosomes look like. Right? At this point, we hit. This is not a diploid number. Right? We have too much chromosomes right now. So in order to get that diploid number, the chromosomes need to be split apart again. And that's why we go through a second meiosis. Right? So the steps essentially repeats itself with one difference. Anyway, any difference in meiosis two? What happens or what doesn't happen in meiosis two that happens in meiosis one? Right? Crossing over doesn't occur again. So in meiosis one, we had crossing over. Meiosis two, there's no crossing over. Right? So in meiosis two, we started back with prophase. Okay. Now, meiosis two can either start one time or you can have a little bit of lag. Right? But the lag shouldn't be too long because you don't want to keep your cells like this. Right? So you can either start immediately or start shortly after. So you starting back with prophase. Again, centrioles move into the opposite ends. Right? Centrioles move into the opposite ends. The genetic information thickening, the chromosomes thickening, and then we enter metaphase, right? So metaphase, same thing. Line up along the pole, right? Once you line up on the pole, it can be split apart in the next phase, which is anaphase. So metaphase, line up along the poles, line up along the equator, and then you split apart to the poles. Right? Eventually you reach telophase. Yeah, and cytokinesis occurs. So you have to know both meiosis 1 and 2, but once you can remember meiosis 1, it's easy to remember meiosis 2, because it's like a repeat step without crossing over. Right? And this happens twice, right? Because remember, we have two cells this time. Okay, so it's occurring in both cells at the same time, or simultaneously. Right? A good way to remember it is to remember the key stages, and then associate what's happening afterwards. So what I mean by that, right? So in prophase, we know we do it for meiosis 1. For prophase and meiosis 1, we know the most significant events, or the significant events of prophase. At the end of prophase, I should have crossing over. So prophase, you can say prophase 1. So, right? The most significant event, I should have crossing over. Right? I should have crossing over, and I should also have thickening of chromosomes, right, to become visible. Right? Again, there are other things occurring in prophase 1, but it's not the main things. It's important, it's just not the main part of prophase 1 in meiosis. Right? The next thing is crossing over to form recombinants. Form recombinants. Right? And then prophase. That's prophase. And then metaphase. The main thing with metaphase, right? So you can remember metaphase, or why I used to remember it. Metaphase starts with an M. What happens in metaphase? Movement. Movement where? Along the equator. Right? So the significant event in metaphase is moving along the equator. Right? In anaphase, right, which is the next stage, we have the homologous chromosomes. Homologous chromosomes being pulled apart to the equator. Now, you're not going to be answering the questions like this, right? It's just a way for you all to remember. If you'll have to answer the question, what happens at anaphase 1? You're going to state the homologous pairs of chromosomes are pulled apart, and it's telling me what's pulling apart by the spindle fibers, right? Towards the equator. Right? So to the equator on opposite ends of the cell. Sorry, not to the equator, to the poles. To the poles at the opposite ends of the cells. Right? And then we have telophase for cytokinesis and separation occurs. Cytokinesis and separation of cells occur. Right? And because this is the end of telophase 1, these cells still contain a diploid number. Cells still diploid. Okay, and then for this, you repeat the steps for meiosis 2, with difference being no crossing over in proteins, and the amount of cells produced would be 4 in total. It's a haploid. In the document, it's have a definition for meiosis, and it explains what happens in meiosis. So one time they do a table, compare, just like a quick table. There don't be any fancy table or anything. So we compare mitosis and meiosis. Right? And when you're doing a comparison at this level, especially in the table form, you're not just writing meiosis and mitosis. Right? What you should also have, if they ask you to draw a table, is a category. So for example, a category here would be number of chromosomes in daughter cells. So it's always good whenever you're doing a table, not just to answer questions, but if you're doing revision, have a column for category. Right? So you know what you're comparing, and whenever you answer any question, whoever's marking it would know exactly what you're comparing. Right? It's a good way to format your answers. Right? So like this. So what's the number of chromosomes produced in meiosis? 23. Right? And in mitosis, it'd be 46 in humans, right? Right? So especially talking about humans here specifically. Right? And if you want, you can put it 2n, you could put n. So this is like just go through a few factors. Right? Or a few categories. So that is number of chromosomes. Right? Then after you compare chromosomes, you compare number of daughter cells produced. Right, so what's the amount of daughter cells produced in meiosis? 4. Right, and in mitosis, 2. Right? And next category could be variation. In meiosis, what kind of variation we have? Well, is variation present? Right? So variation occurs due to crossing. And in mitosis, what kind of variation we have? No variation, right? Genetically identical cells. Okay, hormones. Variation needs to cross over. And in mitosis, we have no variation. The next category association between homologous chromosomes. So the only time the homologous chromosomes come together is for the purpose of exchanging genetic information. Right? So based on that, what would homologous chromosomes associate with each other in meiosis? Yes. Right? So they associate during meiosis and they do not associate during mitosis. Because again, no crossing over occurs in mitosis. So your homologous chromosomes are not going to come together. They're not going to associate. No association. So when you compare them, you're not only talking about the end products, meaning amount of cells and chromosome number of cells, but you're also talking about the actual process. But within the process is different. And one thing I want to mention, because it could come, if you don't come in the paper too, come in the multiple choice, is how to determine the amount of possible combination for a gamete. So for example, right? If you're given a question like, if a diploid cell 2n is equal to 6, divides by meiosis, how many different combinations of chromosomes would be possible to find in a gamete produced by this cell. So again, recall a gamete just simply means your sex cell, right? So if you starting off with your parent cell is a diploid cell. 2n is equal to 6. But remember, our chromosomes, the genes on the chromosomes recombine all the time. So what they're asking for, right? They want to know how much chromosomes would be possible, how much combinations would be possible if this organism has a diploid number of 6, meaning 2n is equal to 6, right? And it have a simple way to do this, or simple formula, I should say, right? So to find number of possible combinations of a gene is simply, all you do is 2 to the power of n, right? So 2 to the power of n, where n, so this, again, sure it come in the multiple choice if it doesn't come in the paper too, or it might even come in the books, right? Where n is equal to the haploid number. Again, this is for meiosis, right? Because remember, in mitosis, we're not going to have any recombination or pairing of homologous chromosomes. Right? They'll line up along each other, but they're not going to pair. Right, so how do the homologous pairs arrange themselves in mitosis before we can answer this question? Right, so if you look in meiosis, remember the homologous pairs are literally next to each other. Right, so this is one homologous pair, this is the next one. In mitosis, they line up above and below, right? In single parts, think about it like that. Right, so let me just go back to the diagram. So this is meiosis, look how they doubled up. Homologous pairs are together. And in mitosis, they line up in a single file. Right? So back to this. Okay, so for this organism, right? This cell, what would be the number of possible combinations? 8, right? So it'll be 2 to the power of 3, which is 8. Okay, so it'll be 2 to the power of 3, which is equal to 8. So if they give you the diploid number, just find half of the diploid number. Right? So 2 to the power of 3, which would be equal to 8 possible combinations. Anybody confused about any steps in meiosis? Right? So we have to know the steps. Most important thing, we have to know where crossing over occurs and how it occurs, right? So homologous pairs line up against each other, right? They exchange at points called bivalence. The point of exchange, we call it chiasma or chiasmata, and what is produced is called bivalence. Right? So the key terms there, chiasmata or chiasma, bivalence, and well, the crossing over. So remember, on the point of meiosis, right? The point of sexual reproduction is the increased genetic variation, right? And how does that variation come about? We have three ways in which variation occurs, right? So the first one, well, what they have as the first one, I have as the first one, right? Is independent assortment in meiosis. So what is independent assortment? Anybody cross it over? So because of it as 2 separate points, I wouldn't say it's crossing, right? Right. So independent assortment. So we know that we get during the process of fertilization. Half of your gametes half of your genes come from one parent, and half of the other genes come from the other parent. So half from the male, half from the female. Okay? But when we say independent assortment, independent means individual. It's not affected by anything else. And assortment means to arrange. So what independent assortment loosely refers to, right, is the splitting apart of these gametes. So that's way you can explain it, is by doing a probability tree. Everybody here do odd maths? Do odd maths? This wasn't maths, so it probability tree wasn't maths? Yeah? No? Okay, it's simple, it's simple. So independent assortment. Right, so it kind of deals with probability. So let's say we starting with an individual, right? they would have a certain makeup of genes, right? Their chromosomes would have a certain makeup of genes. They would have gotten those genes, half would have been from their mother and half would have been from their father. And before that, it would have been the same. But let's say we starting with this person. Right? This individual would pass on their genes, which is a combination of both their parents' genes, to their next generation, right? But in order to do that, you need another individual. Right? So if you think about it, there's 4 sets of genes we deal with. Okay? 2 from this person's parents, and 2 from 2 separate pairs from this person's parents. Okay? So when you pass it on to the next generation, right? So you have 4 sets. For simplicity's sake, I just wrote 4 chromosomes. Right? This is from, show you different colors. There's 4 different sets. What you have to remember is that each person could only hold 2. Okay? So this 2, let's say that's from the dad, this 2 is from the mom. Okay? So that was, let's say these 2 are carried on. Right? These 2 are carried on to this person. Right? Is it guaranteed that these same 2 genes will be carried on to the next generation? No. Right? It might need be carried on at all because that person have to reproduce with another individual. So what independent assortment is referring to is that the transfer of gene is not dependent on other genes. Independent assortment, right? It is a random process. Okay? Independently assorting or arranging genes, right? It is not dependent on any other process. Okay? It is an isolated process. And this comes about because of the difference in gametes from paternal and maternal sources. So if you had to define it, the independent assortment is a independent assortment refers to the So that's one source of genetic variation. If I have a diagram here, this could be easier to explain them. Let me look at the diagram. Right, so if you had to assign a stage for independent assortment, you'll assign it to metaphase, right? Because this is where the homologous pairs line up along each other. Right? So this is after crossing over. If we look at the diagram, we'll see a difference in color. That's usually how crossing over is indicated, right? So originally, this red chromosome it was originally all red, indicating it was from one individual, right? But we see a speck of blue. This blue came from the other chromosome, right? After crossing over. You can't see the red part on the blue, but it did. Same thing with the other pair of chromosomes. Okay, the red part has a bit of blue. This blue chromosome has a bit of red. That indicates that crossing over occurred, right? So for independent assortment, we have to understand that both these chromosomes came from a different source, right? One is maternal and one is paternal. Now, based on how they are arranged, that will determine the features produced in that cell. So for example, right? Now you say the red chromosome has the gene for brown eyes. Yeah? And the blue chromosome has the gene for blue eyes. We're just thinking about eyes for now. Yeah? What will happen is based on how it crosses over, how the chromosomes are split apart, that will determine the genetic characteristics for that cell. Right? Now, obviously, this cell not presenting any of these characteristics. But what it does, it will present the characteristics if it successfully fertilizes something. Right? So what you need to know about independent assortment, it occurs in metaphase. And the basis of it, you have two sources of genes, maternal and paternal, and it's randomly determined, right? Based on the alignment of the chromosomes or the homologous pairs. Right? The next source of variation would be crossing over. And we went through this already, right? Is the exchange between your chromosomes to produce different or a variation in chromosomes. Right? So two homologous pairs are line up against each other, and genetic information is swapped at the bivalent. Right? So you have two reds. This is the point of crossing over. So 2 points of crossing over here. Yeah, this is one. And this is the next one. There's 2 points. So you have two chiasmata. Right? And that will produce this, right? Which is genetically different, right? It's variated. And the third one is random fertilization. Okay? So again, fertilization is the fusion of two gametes, right? So in humans and mammals is when the sperm successfully reaches the egg and exchanges genetic information, right? But again, remember in males especially, there are millions or billions of gametes, right? And all of these gametes are genetically different. Okay, so when we say random fertilization, it means the endless possibility of combination of genes from that parent, right? Or to produce an offspring. Okay, so the main principle in random fertilization, each gamete has a unique combination of genes. Yeah? So while every human is different, even every sex cell, every gamete is different as well. They're not going to be the same because of processes like crossing over. So these are the three sources of genetic variation. A question come and ask for a source of genetic variation. You choose any one of these that you can describe the best. So I have some questions. If anybody questions on meiosis on the next file, right? Well, I want to go through Mendelian genetics first. Let me do the questions. So one, I'll pass the two questions. Quick one. So you give one, the instructions that these three stages represented the same stage in minuses 1, 2, and 3, right? So minuses 1 and 2 and mitosis. Right? The three stages are the same stage right now. The first thing you want you to do is to identify the stage. What stage is this? Metaphase, right? They line up all of the equator. Right? So this is metaphase for all of them. And we figured that out because of the position, the line up all of the equator. That is metaphase. Right? We went to the equator. Now again, I just want to preface, it could be horizontal. Right? Okay. So now the next part of the question would be to identify which one is minuses 1, which one is minuses 2, and which one is mitosis. Okay? So which one is it would be easiest to identify this? So this one would be Daniel, this is minuses 1. What are these? Right, so remember, if they're both of them are metaphase, right? The difference between minuses and mitosis in the metaphase stage is the amount of chromosomes present. Mitosis, it arranges in a single file. Right? The chromosomes line up along the equator in a single file. This has to be mitosis. Right? And in meiosis, they are arranged in pairs. In meiosis, so this is meiosis. Meiosis. Right? Meiosis 2, remember, you already split apart with the crystal. So you're not going to arrange in pairs again. Right? We go on to, so I'm using new documents that it's sent to you. We'll get to the questions afterwards. Everybody here comfortable with like a Punnett square? And like, okay, not just the Punnett square, but like identifying the gametes from before you even do the Punnett square. So if they give you the physical characteristics, you could come up with the gametes. Yeah? Okay. So when questions come on these, especially if you have a chi-square, or if it have even a cross, it'd be a lot of lines. Right? So just we had to be good with this. Because if a question does come, we're going to be for a good bit of months. So these are just some key terms here on the first page in which we need to understand. Right? We need to know what a gene is. And again, a gene is simply a nucleotide sequence which determines the formation of a protein. Right? Based on the coding of that gene, RNA comes in, mRNA comes in, transcribes it, carries it to the ribosome to be translated. Right? And allele here is just a different form of a gene. So if you want to think of an example of an allele, think of icon or HER, right? The gene for HER, the gene is HER. The different alleles are the different types of genes in for that HER. So you could have blonde hair, we could have black hair. Black hair would have a certain allele. Blonde hair would have a certain allele. But both of them falling on the same gene, right? On the same locus of that chromosome. Right? So again, alleles are just different forms of the same gene. Right? We would know what dominant and recessive is. Okay, a dominant allele, right? So genes are not dominant, alleles are dominant. Okay, a dominant allele refers to an allele that would be expressed in a heterozygous setting. Okay, remember, heterozygous means two different genes. On A and on A, this is a heterozygous combination. Okay, this is hetero. Hetero means different. Right? A and A are both homozygotes. Right? Now there are different types of homozygotes. This is homozygous recessive. This is homozygous dominant. Right? Okay. So in a heterozygous pair, whichever one is expressed is referred to as dominant. Usually in bio, the capital letter represents the dominant allele. And for a recessive allele, it's whichever allele that is hidden or not expressed in a heterozygous setting. Okay, so in this case, the recessive allele will be A. Recessive means it is present in your genotype, okay, but not expressed. Okay? Recessive alleles can be passed on to the next generation, but is not expressed by whoever has the combination. The other terms we're going to get to as we go through the topic, right? But so genotype is the genetic combination, right? Your genotype is not visible, right? Your phenotype is the observable characteristics that your genotype codes for. Okay, so your genotype might be capital B, common B, but the phenotype for that would be the hair color, right? Or it could be hair color, eye color, anything like that. Any physical observable characteristics. Right, so it had this fellow called Gregor Mendel, right? And he was a monk back in the day, and he had an obsession with peace. So what he was trying to do, he was trying to see what gives peace its characteristics, right? So remember back then, it didn't have microscopes, it didn't have access to information and stuff like this. So he had to conduct these experiments himself, right? And he had access to plants, animals, and time to do all these things. So what he would do, his first set of experiments, he was trying to prove that physical traits have something behind them. It's not just random, right? You're not just born with these things. Something influences it, right? So what he used plant roots because he understood plants were living organisms, right? And he understood they were simpler to do these experiments with than animals. And it's a good thing he decided that because these experiments wouldn't work with animals, right? Animals take too long to reproduce, right? So what did he do, right? But he didn't do this. But this is how his experiment would look. He would take two flowers, or two plants with different characteristics. Okay, so this plant had pure breeding red flowers. When we say pure breeding, it means it only contains the alleles for red flowers. Right? And he had pure breeding white flowers. Again, it only has the allele to give you white flowers. Right? Okay. So what happened? In his first generation, so he bred the two, crossed the two, and he got all red flowers. Right? So this should indicate something. Right? What would this indicate? So it's pure breeding, and there's pure breeding, you will carry in the animals only for that field. So he didn't know what the animals are, right? He didn't even know what is an animal. But what he knew is something influencing. So again, pure breeding. Red. And pure breeding, white. This is all he said. You don't know which one is dominant, is it? There's no it. Both of them, it's pure. Okay. Now what we can do, we can just assign a value, right? We can assign a value, let's say R and R. Okay, one dominant, one recessive. Okay, we don't know which one is which. So when you cross them, all the members in the first generation of F1 with red flowers. What does that indicate? Was the only possible way that all these flowers could be if both of them are pure? Was the only way this is possible? The only way this is possible is if red flowers are dominant, so white flowers. And because they are pure breeding, they carry the same value on both members. Right? Which would indicate to us that this red is dominant. So because they are pure breeding, this would have to be R. If this is again pure breeding, and we figure out that white is recessive, the value for white is recessive, this would become another complement. And we know if we cross a homozygous dominant, the homozygous recessive, what will be the genetic combination for all the F1? What will be the genetic combination for all the offspring? If you do the Punnett square, you get it. The first generation only contain red flowers. Okay, but at the Cape level, we don't just stop there, right? We go to the second generation, or the F2 generation. So to produce the F2 generation, what he did, taken two of these red flowers and cross them with each other. Right? So you cross this red flowers with another set of red flowers. Now are these red flowers pure breeding red flowers? Good, right? Because they do not carry the same amount. Right? So they're not pure breeding. So what will happen? Okay, no. R is explosive is R common. Will reproduce capital R, capital R, capital R common, capital R common, 2R. Okay, what type of phenotype is it? What do you think of it? Right? So you have two generations, you have one red, phenotype, right? And a silver phenotype. So you have one red, three reds, and one red. So the point of this, the point I'm trying to indicate is when you get in questions or when you do a genetic process, it doesn't know exactly which one is dominant. You need to figure it out. To figure out if something is dominant, you have to cross two pure breeding types. For the trait you're trying to understand. So for example, right? If I want to see which trait for eye color is dominant, okay, between brown eyes and blue eyes. I need to get two individuals pure with the pure breeding state promoted. Okay? Right? And which they produce offspring, whichever eye color that offspring have, it indicates that color is dominant. Or that eye is dominant. Right? Anybody know it's called that type of cross? Or the two types of crosses. So this is an example of a monohybrid cross, right? A monohybrid cross, you're only comparing one characteristic. The characteristic here was plant color. Okay, we also have dihybrid crosses where we compare two characteristics. But for now, we're doing the monohybrid crosses. That's a bar cross or test cross. Okay. Right? You're trying to the test cross is you're trying to find the identity of the generation before it. Okay, so what usually happens, you have a heterozygous, or a plant with heterozygous traits, and then you cross it with a homozygous recessive. What you're trying to do, you're trying to figure out the identity of the generation. That's what you call it a test cross. Right, so Elon, how you describe this combination? Like, how you put wings. So it's capital R, capital O. How you describe it? Describe it. Okay, that's one way, but using like, so you're correct, right? But like using with homozygous, heterozygous. So you describe it. So is this homozygous or heterozygous? Heterozygous. Right? So, right, so, how do you describe it? Right. So, homozygous for homozygous, recessive. Right. Sanji, can you describe it? Homozygous dominant. Homozygous dominant. So just making sure we have the same stuff because when I explain to them, I know we're going to see this. Right? So now, like I did on the scene initially, we should be able to assign the color from this as well. So this will be red. So once that done, then we press on, it's going to be expressed. It will be red, this will be white. So this should indicate something to us. It should tell us the only time a recessive allele is produced is when it's in its homozygous. Right? It cannot be produced any or it cannot be expressed any other time. Right? And when we cross two pure breeding organisms, the trait is always in a 3-to-1 ratio. Okay? So that's what we see here. We cross two pure breeding organisms, one pure breeding for red, one pure breeding for white, and we get the 3-to-1 ratio. So just want you all to try one, just to get the ones that they get lost, moving on to dihybrid crosses. I'll type it out. The question. So it shouldn't take too long, it's most, at most 5 minutes. Right, so you have two alleles that determine the shape of seeds. It could be wrinkled or round. Martin crossed a pure breeding smooth seed with a pure breeding wrinkled seed. Okay, so what would be the outcome, the expected outcome of the F1 generation? That's the first part. So then he decided to cross two of the seeds from the F1 generation together. So what happened then? Yeah. Yeah, so
Yeah, that's right. Well, we did.
everybody got your, ready? Anybody online got you? So Ariane volunteered to do first, right? And so you do the same, first thing I want to do is, so yeah, you are saying that you try to whichever to use, like you is the answer. Take the whole. Yeah, you got it. And so, yeah, all right, that's correct, right? So again, we cross in two homozygous dominants, right? In other words, right, two homozygous dominants, so we produce a 4-to-0 ratio, meaning all the F1 generation will contain wrinkled seeds. Right, that's the answer. So the answer to the first one is the expected outcome is for 4 wrinkled seeds, right? And 4-to-0. Right? The second generation, because the first generation, all more wrinkled seeds, and the only possible combination would be a 2 generation of the heterozygous. So the heterozygous, the gametes would be one dominant, one recessive, one dominant, one recessive. On the cross section, the 3 wrinkled seeds and one monocyte. This is the phenotype. The genotype or the genotypic ratio is. So remember, this is phenotypic ratio, right? The genotypic ratio the ratio of W. Now, W, comma W, W, comma W, right? 2 comma. If we talk about since the genotype, we ignore any phenotype, right? Because we talk about genotypes. This would be a 1, a 2 to 1 ratio. Okay? Phenotypic ratio indicates on what creates first. So the empirical one, genotypic ratio, talking about the genetic combination of the gametes, right? So when I say this, it's talking about phenotypic ratio. So be careful, you should not bring that up because sometimes the question asks for the genotypic ratio. And some of the students put the same, they think phenotypic ratio, genotypic ratio is the same. Or it could be the same, but it's not necessarily the same one, right? I don't have to just understand the difference. The genotypic ratio is dealing with the computation. So the answer for this one would be you'd expect 3 wrinkles on 1 row and just add the central row. This wasn't a population. And so before we move on to test process and dihybrid process, I want to go through Mendel's 2 laws, right? So the first law was the law of segregation. Segregation. Right, so the 2 laws we have to know is law of segregation and the law of independent assortment. Right? So now the law of independent assortment should sound familiar, right? Where did we get that before? Which topic? In variation in meiosis, right? So variation in meiosis, right? For segregation, okay, this is what the law states. Right? Remember, during the process of meiosis, you form a gamete. A gamete is carrying 23 out of the 46 chromosomes. So that means it's carrying half the amount. The reason it carries half of the amount is because it only carries 1 of it. So for example, right? Every living organism in the somatic cells have 2 elements. Right? It can be capital A, capital A, capital A, capital A, well, you have 2 elements. Right? However, the gametes only have 1 element. So this is a somatic cell. Somatic cell simply means a body cell. Right? So it's not a reproductive cell. It's the somatic cell. The gametes, so like this sperm and ova, would only carry 1 element. Okay, and the reason it only carries 1 allele, remember recombination, fertilization needs to occur. So if it's carrying 1 allele and it fertilizes, it would produce, then you get 2 alleles. So the law of segregation is telling us that gametes only carry one allele at a time. Okay, that's what the law of segregation is. Segregation means to separate. Right? The two alleles in somatic cells are separated or segregated into individual gametes. Right? Eventually, if it fertilizes, then you get back your two alleles per locus or per gene. Right? The second law is your law of segregation. Right? And again, you should be familiar with this. Right? It's the same thing from meiosis. Right? So when we're talking about segregation, we're talking about independent assortment. That's what we do now. Meiosis. So the independent assortment, again, we're talking about the recombinants. Right? And how it does, it is not affected by other factors. It is an independent process. Right? And if you had to refer to a stage, you would say it occurs at metaphase. So the combinations that are obtained, they're not influenced by other factors. That's what independent assortment states. So before we move on to dihybrid, let me test cross. Any questions so far on monohybrid crosses or to determine Mendel's laws? So let's do test cross. Right, so why would you use a test cross? Right, so you use a test cross when you want to determine something. Right, so in the name, test cross, you're testing for something. Right, and we use a test cross when we, or back cross, when we want to figure out the genotype of the parent generation. Right, so your F generations are your offspring generations, successive offspring generations. The generation before that is called a parental or parent generation. Okay, so you would know the genotypes of the offspring generation, the F1 generation, but you don't know the genotypes of the parent generation. That's what you want to figure out. The only way you can figure that out is by doing a test cross, or in other words, a back cross, right? Like you're backdating something. So how do you do a test cross? So the one constant with the test cross, it must contain a homozygous recessive. So a test cross is done against a homozygous recessive gene. Okay? The other member is what we tested against. So the other member will be gained from the F1 or F2 generation. Okay, and we do a test cross with a homozygous recessive. You have to use this in a test cross, right? Or backwards. So which one we use? But use it actually. Right, so say we have a peer and it's expressing wrinkle seeds. So what are the possible genetic combinations for wrinkle seeds? Wrinkle seeds could be either heterozygous or it could be homozygous. Right? So think about it like this. If you have a wrinkle seed, right? You just randomly pick up a seed at the grave. Would you know if it is dominant? So would you know if it's homozygous or heterozygous? You wouldn't. So what you have to do, you have to test cross it with a round seed that you know is homozygous. Okay, so you test crossing it against the recessive homozygous. Okay, so it's two crosses you have to do. You have to do W. But you have to do right? So you take in the P, what you're doing is you take the P and the cross, and it's you actually do the cross, right? But that's the experimental part. The theoretical part is what we do. So the possibilities here are one, if you have to assign that test, you will be like Y, right? Because W, capital W, common W, like it's big and small. That's why you use E, things like that. Right? We have two different possibilities. Okay? If, remember, this is what we focus on. Right? If it is homozygous dominant for wrinkle seeds, we're supposed to get in the next generation when we breed our seed with the homozygous recessive seed, we're supposed to get 4 wrinkles. Okay, so we actually do the test, right? Take this seed. Oh, sorry. Right? So if we do, if the identity of our unknown seed is homozygous dominant, we're supposed to get 4 wrinkle seeds. If the identity of our wrinkle seed is heterozygous, we're supposed to get 2 wrinkle seeds. 2 wrinkle to 2 rounded. Right? So in other words, a 1-to-1 ratio. You understand? So you are actually performing the test. However, based on the results of the test, you can figure out, okay, what is the genetic identity of that organism. Right? So let's say we do the test, we do a lab, we do the test, and we get a ratio of 1 wrinkle to 1 wrinkle. That means that our offspring, sorry, our organism was heterozygous. The test cross is used to determine the genetic identity of an unknown or of an unknown genotype. Right, and if you know the identity of this organism, you can figure out the identity of the parental. So that's what's stated here. If the offspring all shows the dominant trait, then the parent must be homozygous dominant. That's the only way you could get all offspring with the dominant trait. Because remember, we are crossing it with a homozygous recessive. That's the means that I want you all to take a test cross, you cross against a homozygous recessive, a known homozygous recessive. Right, and based on the ratio, you can determine the identity of the parental organism. Any questions on the test cross? Okay. So you always see this term, right? A term called wild type. Anybody can tell me what wild type means? So when they say wild type, it's simply referring to the dominant allele in a population. Okay, so for example, let's say we have a group of flowers, right? It wasn't influenced, it wasn't crossbred with human influence. And those group of flowers, the majority of them are colored yellow. Okay, the wild type allele would be the dominant allele. So the allele that codes for yellow would be considered your wild type. Okay, so when I say wild type, I'm simply referring to the dominant allele in that population. Okay, now it would have variation. Some might be purple, some might be blue, but the wild type is the most dominant, the most common characteristic within a population. The dihybrid processing. So it's in the name, right? A dihybrid cross. The monohybrid cross is comparing one characteristic. A dihybrid cross are comparing two characteristics at the same time. Right, so we're sticking with these. So this time we do coloration and texture or shape. So these are two different characters, right? Now it's out of 4 in total, but these two characteristics found on the same look of the gene, right? Meaning these two would carry certain alleles based on whichever one dominant to be expressed. These two characteristics are found on the same look as well. These two, this characteristic influences this characteristic. Right? They found these two found the same loci, but not influencing this. Right? So meaning, if a P is yellow, it doesn't have to be rainbow. If a P is yellow, it doesn't have to be sweet. It doesn't independently assorted. In this case, lots of exceptions. Right, what information I need to give you? I need to tell you which one dominant. I'm telling you that soon, in this case, right, dominant. So wrinkle, so different from the last ones they gave you, right? In this case, food is dominant to wrinkle, and yellow is dominant to wrinkle. Now we're going to do a diagram first. Can we use the same letter for both characteristics? No, it wouldn't make sense. We have to assign different letters. Right, so let's stick with the W for this. Okay, I'll take that system. And we'll use minus. Right? Okay. Now, with dihydric process, you want to compare two characteristics at the same time. Okay, so we have to put them together. But alleles, they come in pairs, right? So each one have to come in pairs. So for example, what I would do, I would give you only two I'll give you only two pairs. So cross the true rooted string. This is the next way of saying it, homozygous dominance. Everything. That should be that simply means homozygous dominant for both traits. Right? So we cross the that tutor, homozygous. That's for both traits. And that's all the information I'm giving you. We can figure out everything we need to figure out. So again, true breeder means that homozygous dominant for both traits. Now you figure out the genotype. What will be the genotype for our true breeding strain? Obedient type. Abishal. Abilasha. Sure. Abilasha. This. They were also discarded so far. But remember, we also have to account for the yellow and green. Right? It's a diagonal force. Right? So true breeding should mean yes, it will be dominant for smooth, homozygous dominant for smooth. But it will also be dominant, homozygous dominant here. We have to account for both loci. Each loci has two elements, right? One from parent, one from mother, one from the father. Right? So that's what we call homozygous recessive combination. Yeah. The dihydric cross, we're not going to get a ratio. We're not only going to get 4 species. We're going to end up with 16. Okay? Sure. So anybody still talking with this? So when we did the monohybrid cross, because we are accustomed to that kind of cross, we skipped a few steps, right? So you can see that you would have had to write the gametes and then do the cross, right? We're going back to that zone. But no, because we have a good bit more gametes. We're going to put the gametes. So we need to find all the possible gametes of the true relation. So what are the possible gametes? Right? So in monohybrid, we just write S, right? They come in pairs. So you can't do that. Each gamete here have to have 2 letters. Right? And how we get the letters? You just look at only possible combinations. Right? So the only possible combination in this case, because it's true breeding, it can only be capital S, capital 1. Right? If it was different, what you're supposed to do? You're supposed to do this. So it's across everything. It's like in maths when you're doing when you're multiplying brackets. You multiply everything by everything. But in this case, we just write one in the gametes. Because it don't make sense to just repeat multiple times. If they want to do it twice. You know, write it 4 times. Same thing with homozygous recessive homozygous. Right? To get the gametes, you have to multiply all the elements together. You get this one. Again, you don't have to write it twice, but writing it twice. And then we just cross them, right? So we can just cross. That's the only way. Doing this cross like this. So as so the gametes, remember the gametes over here, right? So the SY as the top part. Right? We have common as common one. Commonness, common. And this you cross it like this. Right? So S, so this is how you do it. You multiply the related letters. Right? So S capital S by commonness is capital S commonness. Capital Y by common one is capital Y common. Because all of them are the same, we're going to simply not in this case. Right? This is the F1 generation. The F1 generation is, so what are the phenotypes here? So the phenotype for this capital S common S is smooth and yeah. So it's smooth, yeah. Right? We get 4 yellow smooth or smooth and yellow peaks. And the F1 generation. Right, so 2 characteristics we obtain. We're going to do our F2 generation. So for our F2 generation, we cross 2 members from our F1 generation. Okay, so what are the numbers from our F1 generation? Capital S from 1S, capital Y from 1 times capital S from 1S, capital Y from 1. And again, both of them are phenotypically smooth and yellow. So this is what I was talking about before. So we have to figure out the gametes to figure out the F2 generation. Right? Figure out the gametes, right? What are the gametes for this one? So the first possible gamete, remember these are just possibilities. S by Y. Capital S, capital Y, that's the first gamete. Capital S by common one. So for the second possible gamete is capital S common one. Then we move on to the second S. Common S by capital Y. So common S by capital Y can give us a common S, capital Y. And then common S by common Y. So we can also have common S, common. Everybody saw what did that, right? No, because this is the same thing with the same possible gametes. Right? S one minus one. Everybody understand how to obtain my gametes? Yeah. So this is the part I was talking about. Now we have 6 possible combinations. Thanks. One thing again. So to do this, we just take any gametes from one parent and we crossing it against all the gametes from the second parent. Because we have 4 different gametes, we need to have 4 different columns and rows. Right? Hence Y, hence Everybody understand how we get up to this point, right? And then we just cross it, like capital S, capital S, capital Y, capital Y. S capital Y, Y by common Y will be common Y. S by common S. So this one we go. What? These are two heterozygotes, heterozygotes, right? Meaning, dominant recessive. There's two different elements. Whenever you cross two heterozygous organisms, you're always going to get this ratio. Anytime you cross two dihybrid heterozygous organisms, you're going to get a 9 to 3 to 3 to 1 ratio. Okay, you're supposed to get 16 different, well, 16 combinations. So this adds up to 16, 9 plus 3, 12, plus 3, 15, plus 1, 16. Right? Now, some of this is the genotype combination, right? The phenotypic combination might or may not be easy, but it's the combination of it. Right? So I'm just going to put some randomness. But this one here, what will be the phenotype? Right? It is heterozygous for smooth and wrinkles, so that means it's smooth. And it's recessive to be colored. So that means it will be smooth wrinkle. So right, so 9 to 3 to 1 ratio is the ratio obtained. So what you have to do, you need to pick out the phenotypes that each one is. So smooth, smooth, smooth. All these here are smooth in the first row. Right? This smooth. All these in second row are smooth. We have 8 smooth so far. We have 1, 2, 3. It's just a ratio you obtain after the one function. Right? You always get this ratio when it comes to heterozygous organisms and a dihybrid. It's a dihybrid cross. It's not more difficult, it's just more weak. Right, so we come to the end of this. So we're nearly done with genetics calculation. So what we have to do is the chi-square test. Before we do the chi-square test, we need to know about the null hypothesis. All right, so a null hypothesis, right? So whenever we do an experiment, we have a problem statement, right? We have an aim and we also have a hypothesis. Okay? If it's a plan design now. You all did a genetics lab in school? Everybody else? No? Okay. So what you'd have to do, you have your normal hypothesis and then you have a null hypothesis. Your null hypothesis is basically the opposite of your hypothesis. Remember, hypothesis, the point of a hypothesis is to prove or disprove. Right? You don't have to be right in a hypothesis. Okay, so when you have a null hypothesis, again, you're trying to prove or disprove it. Okay, so for example, a good example. We use the example here. So you have a study to look at the relationship between sleep deprivation and test performance. Okay, so you have a hypothesis stating that study is designed to this study is designed to assess the hypothesis that sleep-deprived people will perform worse on a test than non-sleep deprived people. Right? So the more you sleep, the less you sleep, the less you explore the test. That's the hypothesis. The null hypothesis, right, would state something opposite to that. Right? I'll write this so you just write something out for this. As an introduction. Right, so you have null hypothesis. So the hypothesis is that those who are sleep deprived perform worse on the test. The null hypothesis would be the opposite. Those who are sleep deprived would not perform worse on the test. You understand? Right? So the hypothesis stating something in null hypothesis going against that. Because you want to prove or disprove one of them. Right? And that's important for the chi-square test. Because when we're doing the chi-square test, we're not proving or disproving the hypothesis. We're proving or disproving the null hypothesis. Okay? So this table looking familiar. This table should look familiar, right? Is a probability. It helps us with probability. So what the chi-square test is, it's dealing in terms of probability, right? So how it works, okay. If something has a chance, is 0.05 here represented 5%. Okay. The principle behind the chi-square test is that if you have a 95% chance of something occurring, then that's a good thing. You have a high chance that you're right. Don't have a chance you're wrong, but you have a better chance that you're right. If you have something less than a 5% chance of being right, you're not going to accept it. Yeah. What you all need to know, you all need to know the principle. What you all need to know is if the value you obtain in the chi-square test is lower than this value here, then this 3.84, you reject the null hypothesis. If the value is higher, you fail to reject the null hypothesis. Okay? Fail to reject means accept, but we state it as fail to reject. Right? So I get into the actual calculation. Right? After this, I'll do the question one time so you can understand it better. So we have green and yellow seeds, right? Or green and yellow something, whoever it is. We say seeds, green and yellow seeds. So we have a bunch of them. We have a total number of 880. So we have 880 seeds divided into green and yellow seeds. If we count them up, we'll get 639 seeds. If we count up the yellow, we'll get 241 seeds. Okay? Now, that is our observed value. Meaning we do the test, we do the count, and this is how much we observe. Right? We have an expected value. Okay? So this expected value would come from our crosses. Right? When we do the question, you'll see how we come up with the expected value. But for now, the expected value comes from our cross. From our ratios, that's how we get the cross. Right? what we need to do is have a formula for chi-squared. Is this formula x squared refers to chi-squared. So the formula for chi-squared is the difference between the observed value and the expected value squared over the expected value. Okay? This vocabulary. Terms, this chi-squared is equal to the number of observed minus expected squared. Right? So observed, again, I know it's a different table, but if this is the amount of observed in the first row, what we are doing, right, we're subtracting observed, which is 129, from 135, which is the expected. Right, 129 minus 135 is -6. And we square that number to end up with 36 over 135. Right? And you're doing that for each different category. So one category was yellow, one category was green. So we're doing that for both. For green, it was 51 in the observed minus the 45 from the predicted. Right? And you square it. And you divide by the amount of predicted for that category. Okay? And you get a chi-square value. Now, this chi-square value, you have to use it and compare with this table here. So you'd see it have two columns, degrees of freedom and probability level. Right? So the question would give you this. It would give you the probability level. So it would tell you if to look at 0.05, if to look at 0.01 or whatever. At the CAPE level, most of the time, they tell you all to refer to the 0.05 column. Right? The only thing that would be different is dependent on your degrees of freedom. Right? Degrees of freedom, to calculate this, you're looking at how much categories you have. How much categories we have here? 2. So to calculate degrees of freedom, it has a formula that n, which represents degrees of freedom, n is equal degrees of freedom is equal to n minus 1. So if you have 2 categories, it will be 2 minus 1. So you have 1 degree of freedom. Right, so what you're doing is you're going to the row with 1 and you're comparing it with the row right under the probability of 0.05 or 5%. And that is the probability value that you're looking at. And so remember, our chi-square value was 1.0667. The probability level we're working with is 3.841. Which one is less? Our chi-square value or the probability level? The chi-square value. So when the chi-square value is less, we share it back. When the chi-square value is less, when the chi-square value is less or lower, we reject the null hypothesis. Right? So whatever our null hypothesis was, we rejected it. Because when we calculate the probability, it was lower. Right? I know that just based on talking, it'll be difficult to understand. But we're going to do a question now to help you already understand. Right. So the probability table is give you all that in the either in the question or the data book, right? So you'll have to memorize it. Okay, so look at this question here. In a cross between 2 types of pea plants, one heterozygous for the round yellow seed condition, and the other one pure breeding with wrinkled green seeds, the following offsprings were recorded. So what category would this fall under? Observed or expected? Observed, right? So these would be observed. So they want us to clarify the sequential steps when taking the chi-square test to determine the significance of the results. Right? So when they say telling us to determine the significance of the results, they're telling us to accept or reject the null hypothesis. That's what they're asking us here, right? So the first thing we need to do is our cross. Because we need to, if we have our observed, we need to figure out the expected. Right? We need to figure out the expected. Okay, and how do we figure out the expected? We need to do a cross. So we have one heterozygous for round yellow seed condition. And again, you need to understand that what that means. So if you are heterozygous for round and yellow, right? What are the dominant traits here? Round and yellow, right? In a heterozygous organism, whatever traits are expressed, those are the dominant traits, right? So we could start right this one time. For the round, right, we use capital R, slash R, it will be round, capital R will be round. Common R would be wrinkle. And with yellow is dominant color, so capital L is for yellow. And common L is, so common Y is, yeah, right? So everybody understand how we come up with the key, right? That's the key part. Right. So let's figure out the genetic identity of our organisms. So the heterozygous round yellow seed, what would be the genotype? What would be the genotype of our heterozygous round and yellow? Capital R, common R. Capital Y, common Y, right? And then we have the crossing that with a, so then they say a pure breeding wrinkle green seeds. Is this dominant? A pure breeding, but is it pure breeding for dominant or recessive? Recessive, right? So all would be capital letters, right? So we RR, common Y, common Y. And then we do the cross. Before we do the cross, we need to get the gametes. So it'd be R, common R. That's one. It'll be Y. Sorry. R, capital Y, capital R, common Y. Common R, capital Y, and common R. Common Y. So we have four possible gametes, right? Now remember, each gamete have to recombine with each other. So even though it only have one possible gamete for the homozygous recessive, I still have to use four of it, right? So it'll be RY times 4, or 4. Yep. Right, so then we add the cross. No, this is not a dihybrid cross we do before, because it's a heterozygous against a homozygous recessive. Right? So I'm going to do it on the board. Did they want this question to stick on? That would be cross, right? So capital R, common R, capital Y, common Y, capital R, common R,
capital Y, capital Y, capital R, common Y. That would be our diagram, right? Right, so again, we have 16 combinations here. We need to figure out the ratio. Right, we're doing phenotypic ratio. So what we need to figure out, we could only get 4 types of peas, right? What are the 4 types of peas? We can have smooth, wrinkled, which is the dominant type. Right? We can have, I'm just going to write the sentence. We can have smooth wrinkled, or we can have smooth yellow. We can have smooth green. Right? We can have wrinkled yellow, or we can have wrinkled green. Right? Those are all possible phenotypes. So let's figure out how much are smooth yellow fruits, right? So capital R, common R, capital Y, common Y. Which phenotype is that? Wait, remember, we based it based on the only one, right? So I just write R is for capital R is for row, and common Y is for so smooth, so capital R will indicate that it is.
Yes,
So this will be round and young, right? So far we have 1, right? And if you notice, this entire column is round numbers, right? So we have 1, 2, 3, 4 so far, right? Okay, what this column will be round green, so we have 1, 2, 3, 4 so far. Whereas that column down, what would this column? Go the other, so it'll be 1, 2, 3, 4. That's the entire column. What is called? So what the ratio? 1 or 4 to 4 to 4, which is 1 to 1 to 1. Right? So this is our expected ratio. Right? So we have our expected ratio of 1 to 1 to 1 to 1. And this is expected. Now we also have our total value. What we need to do now, we have the ratio for expected, but we need to assign a number to that. Right? How much number of each category would we expect? Right? So we split up in 1 to 1 to 1 to 1. Right? So to figure out the number of expected, we need to find the total amount of peas and divide it. How much we divided it by? 4, right? So all we're doing is we add up the numbers in the ratios. 1 plus 1 plus 1 plus 1 is 4. Right? In this case, we divide it by n, which is 4. So the total number, right? We adding up 108, 108 plus 102 plus 105 plus 101. Right, so the total number is equal to 416. So we divided 416 divided by 4. And we get 104. Right, so because it's in a 1-to-1-to-1 ratio, it means each category is expected to have 104 peaks. Okay, right? So we multiply each category, its ratio, by the number of expected. So we 1 by 104 for each one. So the expected is 104 each. Okay? Next thing we have to do is find degrees of freedom. How much? What will the degrees of freedom be? N minus how much? 4, which is equal to 3. And so again, the degrees of the 4 came from the number of categories. We had 4 categories. So we subtract n from 1, right? And in this case is 4. Right? And then we do our chi-square test, right? So we're doing it. I'm going to put the observed next to each one. So each one we expect 104, 104, 104, 104. Right? So the formula is observed minus expected, right? So we could just subtract for the first one, right? 104, sorry, 108 minus 104 squared. Right, so you would need all the calculators for this one. 108 minus 104 is 4. 4 squared is 16. So we divided, how much we divided that 16 by? 16 divided by the expected, right? How much was expected? 104. So 16 divided by 104 is 0.1538. We're doing it to 4 decimal places. Anytime you're doing this calculation, 4 decimal places. Right? Not 2, not 3, but 4. Right? We're doing it for the second one as well. 102 minus 104 is 2. 2 squared is 4. 4 divided by 104 is 0.0384. Actually 5. Right, we're doing it again. So it doesn't matter if you get a negative value. When you square it, it'll become positive. 0.0096, and the last one would be 0.1538. And then we find in this, sorry, no. 3, 9, 0.0865. Right, and then we have to sum them, right? Right, so anybody sum them, is it? 0.01538. So somebody else could do it at the same time in case I add them. 38. I got 1.0669. Right, so just double check that one, but Yeah, no. Where you go? No. Point zero two eight four. Point zero into two. So it was 0.08. 884. Okay, well, we'll go with it. Right, so okay, we have our chi-square value and we have our degrees of freedom, right? So we go into the table, right? Where do I need to? Right. So we are 3 degrees of freedom, right? 3 degrees of freedom, and we will go with 0.05%. Right, so the value we're working with is 7.82. So we compare the 7.82 to the 0.02884. Which one is higher? Which one is lower? Is our chi-square value higher or lower than the probability? Right? Because both of them low, right? But I'll fix it. That's right. And so in this case, it will matter because it'll still be less, right? So because this value, this chi-square value is less than the probability value, what do? When it is less, we reject. Right? Or when it's lower, we reject. So because the chi-square value, the calculated chi-square value is lower than the probability value from the table, we reject the null hypothesis. In this question, they did not give us a null hypothesis. But if they did, or if we came up with one, we reject it. Right? Everybody understand? Okay. So that's a pretty big part of genetics, right? We still have to do incomplete co and sex-linked traits in genetics, right? Sorry? Yeah, the simplest stuff, right? I just wanted to get through all the calculations, the crosses, and stuff like that, right? But what you all could do to kind of speed up the process and like get some revision. In this same document, it has some questions. I want you all this evening. So along with the graph from last time, I want you all to try this question. Here's a meiosis question. So the one for 21. It's not numbered. In the document, it will be numbered, but it's the one that's talking about meiosis. So describe the stages of meiosis. Sorry, mitosis. We're doing that question. Right? And we corrected first thing tomorrow. And then we'll continue. To describe the stages in mitosis, that question. And we're doing the one label that's 8 as well. Right? Anybody have any questions from the genetics we do today? You all think you all understand it a bit clearer? Right, so those online, you'll have any questions? Good. If you're all good, we'll continue tomorrow. Tomorrow is day 5.